Have scientists created life? This video explores the convergence of cellular synthesis, genetic engineering, and modern laboratory analysis. We look at the benchmarks of synthetic cells, the composition of the SYN 1.0 genome, and the technical parameters required for bioreactor success in this emerging field. What does creating life actually mean? We examine the differences between building an organism from scratch, cloning existing organisms via nuclear transfer, modifying current organisms, or attempting to reactivate dead cells. Each path presents unique scientific, technical, and theoretical challenges. To define life, we look at fundamental biological traits, metabolism and energy exchange, reproduction through the transmission of genetic information, the process of evolution, and the ability of an organism to react and adapt to its environment. The theory of abiogenesis asks how the first life emerged. This involves understanding the conditions of the early Earth, the transition through chemical evolution, the role of natural selection in early biological systems, and the emergence of the first self-replicating molecules. The Miller-Urey experiment remains a landmark study. By simulating the conditions of the early Earth, including atmospheric gases, water, and electrical discharges, scientists successfully demonstrated the formation of amino acids, the basic building blocks of proteins. The RNA world hypothesis suggests that RNA played a dual role in early life. RNA acts as a carrier of genetic information while also functioning as a catalyst, known as a ribozyme, to accelerate necessary chemical reactions. What can scientists build today? Current research focuses on creating simple artificial structures, such as lipid membranes and vesicles, and exploring the basic mechanics of self-replication in these protocells. In 2010, Craiג Ventъr and his team achieved a significant milestone by creating a synthetic genome for the bacterium Mycoplasma mycoides. This was a pivotal moment in understanding how synthetic DNA sequences could define an organism. Was this a truly, quote, artificial organism? There are strong arguments on both sides. Proponents highlight that the genome was entirely designed and synthesized from scratch, while critics point out that the experiment relied on an existing natural cell as a host. DNA synthesis continues to evolve. Scientists are now able to synthesize viruses and simple bacteria, move toward designing complex genomes from the ground up, and iterate on methods to create increasingly sophisticated organisms. Synthetic biology has broad applications. In medicine and pharmacology, it helps produce vaccines, insulin, and pre-programmed bacteria. In energy, researchers are developing biofuels and engineering new metabolic pathways for industrial use. Can we create a cell from scratch? The answer remains, not yet. While we can integrate synthetic genomes into living cells, performing a complete de novo synthesis of a functioning living cell remains a major future challenge. The greatest problems in modern biology remain the core mysteries of life's origin. Understanding the exact mechanisms behind the emergence of the first genetic information, the first metabolism, and the first self-replication. Despite these challenges, much has been achieved. Scientists have successfully designed and synthesized an entire bacterial genome, transplanted it into a host cell, and observed that the resulting organism is capable of replication and function under the control of the synthetic DNA. In conclusion, while scientists have made incredible progress in designing and modifying life, we still cannot definitively prove the specific steps that led to the emergence of the very first life on Earth. As we conclude this overview, it is essential to reflect on the ethical considerations. The ability to manipulate the fundamental building blocks of life brings significant responsibility to the scientific community and society at large. Safety and regulation remain paramount. Establishing robust frameworks for synthetic biology ensures that innovations are developed securely, preventing misuse and addressing potential environmental or biological risks. Looking ahead, interdisciplinary collaboration will be key. By combining insights from chemistry, physics, computer science, and biology, researchers aim to bridge the gap between inanimate matter and living systems. We are entering an era of design, build, test cycles in biology, moving from observing natural processes to actively engineering them. This paradigm shift will redefine our relationship with the natural world. The quest to understand the origin of life is not just a scientific pursuit, but a philosophical one. It challenges our perception of ourselves and our place within the evolutionary tapestry. Thank you for joining us on this exploration of the frontiers of synthetic biology. The journey from chemical compounds to complex life is one of the most compelling stories in modern science. If you wish to delve deeper, the following slides provide a list of recommended resources. This bibliography covers scientific literature, academic publications, and key sources regarding abiogenesis and synthetic biology. You can explore fundamental books by authors like Nick Lane and Paul Davies, along with seminal research papers from the mid-20th century to recent milestones in the field. Additionally, staying informed through reputable online sources such as NASA Astrobiology, Nature, and Science will help you follow ongoing breakthroughs as they happen. We encourage you to use these references to continue your learning journey. Understanding the science behind life is a lifelong pursuit, and these materials offer a solid foundation for your research.